This protocol allows automation of the tracking of growth of animals, using a simple label to identify each animal and a video analysis tool to detect the tags. This protocol significantly reduces the number of hours needed to analyze the video recordings using landscape data, to extract more accurate conclusions from the experimental studies.
Animals in other environments that can be tagged in a stable fashion could be tracked with similar protocols, providing application to chronobiology, ecology, behavioral research, or neuroscience.
We have provided a hardware design and open source code of the image processing model, and we recommend user adopting the protocol to learn Python programming language.
To construct a tag for the target animal, first cut 4 40mm diameter circles from a black plastic sheet and 2 26mm sided equilateral triangles and 2 26mm diameter circles from a white plastic PVC sheet. Mark the center of one white triangle and one white circle, and make a 10mm hole at each mark. Then glue one white shape into the center of each of the 4 black circles. To set up the experimental area, place infrared dark time LEDs into the tank. Keep the infrared lights on at all times. Next, place blue light time LED lights into a modified fiberglass tank containing sand and 4 flexible PVC pipe burrows and connect the lights to an apparatus for managing the photo period. When all of the lights have been placed, position a chilled seawater inlet at one corner of the tank with a corresponding outlet at the opposite corner.